All right. So, as per the last session, we have checked the quality and we require the trimming for that. And for the trimming, we are using the tool called FastP. So, if I will just open the manual of FastP using the FastP space hyphen help and enter, you can see that it is showing you the usage, like how we are going to use the FastQC. So for that, fast P, which we are going to use for training. So for that, it is showing you the usage. So see, if you have a paired and data,
Can
you,
can you share your screen?
oh, sorry, sorry. Yeah, yeah, is it visible now? Yeah, is it visible?
Yes, ma'am.
Okay. So what I'm saying that like yesterday as we discussed the quality control and today we are going to discuss about the trimming part. That how we are going to trim that red color adapter which we get yesterday here, right? Like this adapter, this red color adapter, which is a Illumina universal adapter, and the sequence I have given you yesterday, like this is the sequence. So this we want to trim it from our FastQ file. So for that we are using the FastP tool. Yesterday I have told you how to install the FastP, right? So you just have to see FastP space hyphen help, and when you will enter the if the manual is open, it means the tool is installed. So in the FastP, as we want to trim the two files because we are having a forward file as well as the reverse file because our data is a paired and data. So we are providing the two options here. First, a small I hyphen small I, that is your first input file, which is your forward input file. Then hyphen capital I, that is for your reverse input file. And then again, as it's a parent data, so obviously it is going to generate a two output files, hyphen O small o, that is for the forward output file, and hyphen capital O, that is for the reverse output file. And then we have to give one option, and inside that we have to put the FASTA sequence to trim. So here, this is the option we have to give hyphen hyphen adapter dot fast adapter underscore FASTA. So here in this option, we have to put the FASTA sequence of the adapter which we want to trim, and that we have to put it into one file. So you have to create one FASTA file, and inside that you have to put the sequence which you want to trim, and that file you have to give in this inside this option. Which means like right now we need to create one FASTA file. Okay. So to create one empty file, again I'm using the touch command. I'm opening the terminal from CRISPR folder. I have shown you yesterday how to open it from the CRISPR folder. So you have to select the path and then, yeah, so you can see I'm having this FastQC report and the FastQ file. So you have to just select the folder like this, type the WSL, and the terminal is open. And whoever is using the LTS, I have told you yesterday how to set the path for the LTS, right? So using the touch command, now I'm going to create one FASTA file by the name of adapter.fa or adapter.fasta, whatever you can give, and enter. So once you will enter, you can see here this adapter FASTA file is created, and inside that I am giving the adapter which I want to trim. So FASTA sequence, obviously it is started with the greater than symbol, then some header. So for now I'm giving the H1 header. You can give as per your choice. And this is what the sequence I want to trim. This sequence list I have provided yesterday. So just take the first sequence from it, paste it, and then save it. So this is your adapter file is created. And now we want to create the or we want to prepare the command. So what is our command now? I will just write so that you can understand here. Fast P, we have to call the tool in hyphen small I. We have to provide our input file. What is our input file? This is the SRR ID file which we have downloaded. Like this is our sample FastQ file. You can copy from here as well. Like you can copy from here, right click, rename, and you can just copy from here as well. And then you have to put it. Then hyphen small o, we are providing the output file, the forward output file. So I'm just providing the prefix and then the file name as it is, so that this is our output file name. Now I'm providing the second input file. So in a capital I, we have to provide it. This is our reverse file. So I'm making it 2. And then in capital O, again we are providing the prefix trim underscore SRR ID and then underscore 2.fastq. So this is our reverse fastq file. And then in hyphen hyphen adapter underscore fasta. So this is our option. And inside that option, we are providing our adapter.fasta file. So this is our command ready for the trimming. Now I'm just going to copy this command and then I'm running it. I will just give this command in the chat box as well. So maybe if you want to perform, you can just paste this command first in your script, just how I have created, and then edit as per your system and then run. Okay? I'm sharing this in a chat box. You can give the heading like this, streaming of reads. Yeah, just how I have given. You can copy paste as it is the same command. And you can see here the trimming is done. So once the trimming is done in the folder, by the name of trim.fastq, these files are generated. So these are your trimmed output files. Now, if you want to see really this adapter is trimmed or not from this FastQ file, so what you can do, you can again check the quality. What you can do, just to make sure that now the adapter is trimmed or not, you can again check the quality. You can try with FastQC and you can give this file names trim and this second file name also, reverse file name also. So this is how you can perform. So now if I will just show you, I'm sharing this step as well so that you can try. So whatever the steps I'm sharing it with you, you have to first paste in the chat box and then, sorry, first paste in the script and then run it. I'm pasting it in the chat box. Please go through it. Yeah, so now here the FastQC HTML file is generated for the trim samples. You can see this is what a trim SRR ID underscore 1. And you can see here the adapter, this adapter is removed now. And the per base sequence quality is also better. See? So this is how using the FastP we can trim the adapter and we can just improve the quality of our data. Clear? Any doubt in this step? Uh, so can we proceed further? Adapter file. Okay, fine. Yeah. Can you see, ma'am? I just gave the greater than symbol, then header, and then the sequence. Okay? Hmm. All right. So once the trimming is done, what will be our next step? That we need to align. What is the next step? That is alignment of reads to the reference genome. So for that, we are going to use the tool called BWA, which I have told you to install yesterday, right? But right now we don't have any reference genome yet because this data is from the Homo sapien organism. So we require the Homo sapien reference genome, obviously. But like as we are performing this for demo, and our in our normal system we can't deal with the complete human genome sequence because it required a lot of space, speed, and the time, and we don't have that much time as well as the space, because if you have not that much space and you're running the human data, it can harm to your system. Maybe your data can corrupt, your data can crash. So just to avoid this thing, we are taking just one chromosome from a human, and we are uh, considering this as a reference for now. So this data which we have decided for the analysis, this is checking the variation in a BRCA1. Yeah, in the first session I've shown the SRA page. So BRCA1 gene, BRCA1, this BRCA1 gene is located on 17th chromosome of human. So we are downloading the 17th chromosome of human instead of the whole genome and considering this as a reference for now. So downloading the CHR17 from UCSC database. So I have the link for the UCSC database. It's not like that you have to download it from the UCSC only. You can download it from the NCBI as well. But it's just for like understanding. I'm giving you the link. You just have to run this command on the terminal, which I'm sharing in the chat box. So it is going to download the 17th chromosome. So this wget command is used to download any file from the browser to specific folder by the terminal. So here you can see I'm just running this command, and it is downloading the 17th chromosome pasta file.gz. You can see this .gz file is downloaded here. But as this is a zip file, we need to extract it. And for the extraction, again, we are using the command, then gunzip, and then this file name, chr17.fa.gz. Whatever the commands I'm pasting in the chat box, please copy paste it somewhere. Because later in the group, there are so many messages like we need some commands and all. So that's what I'm sharing in the chat box. You have to copy paste it somewhere. Even if you're not running right now, just put it with you. And whenever you're running it, it will be helpful for you then. Yeah. So this CHR617 is extracted now. Okay. So once this is extracted, now we are going to be align our reads to the 17th chromosome of human. But this alignment contains two steps. Okay, this alignment contains two steps. What is the first step of the alignment? That is creation of indexed genome. What we are doing, first of all, whatever the reference genome we have downloaded, like in our case, it's the 17th chromosome. So we are going to be do the indexing of the reference genome. What is the meaning of indexing now? So like we are having a reads and they are very small as compared to the reference genome. Like they are not, they are very smaller in length as compared to the reference genome. So whenever we are going to directly align the reads to the reference genome, it is going to take a very long time. So what we are doing is we are just dividing the reference genome into a small file, small files, and that files they are in a binary format. So what will happen as we are dividing the reference genome into a small files and they are in a binary format. So tool can easily understand the binary language and it will increase the speed of mapping. So the use of indexing is just to increase the speed of mapping. Means to understand this indexing concept more clearly. Let's take an example of book. In a books, we are having the index, right? So maybe if you want to search a particular chapter, you are going on an index, you are checking the page number, and you are directly jumping on that page number. You are not searching one by one all the pages there. So what it will do, it will save your time and increase the speed of searching. So the same logic will apply over here. It will increase the speed of alignment. It is going to increase the speed of mapping. And that's why we need to do the indexing. So for the indexing also, we are using the BWA tool. Okay, the meaning of indexing is clear. What about others? Priya ma'am, like if you are getting such kind of error, file doesn't exist. There are two chances. Either you are running it from a wrong folder, or maybe the file is not created properly. So just check it once, like the command which you are running in that folder from where the terminal is open, in that folder only you have this adapter FASTA file, or maybe the path is correct or not. Just check it once. Okay. You need to see the terminal, like from where the terminal is open, and other thing like in that specific folder only you have the adapter FASTA file. All right. Hmm. Okay. What about others? Yeah, indexing is clear. Just tell me that because this concepts at least you should know. Okay, fine. So now, as we are using the BWA tool, so let's open the manual of BWA first of all. So if I will just type BWA and enter, so it is going to be open the manual of BWA. If it is installed, then and then only it will open. And here you are having the option of index, like index the sequence in a FASTA format. So to understand it, you just have to type the BWA space index and enter. So it will show you how to prepare the indexing command. So here they have provided the usage like BWA, then option we have to give index, and inside that, then you have to give the option hyphen small a, which is your BWT construction algorithm. Means what? This is your indexing algorithm under the hyphen small a. But in the downside, if you will see, there are two options of hyphen small a BWTSW, and hyphen small a DIV. So this BWTSW, basically it does not work for short genome. Okay, it does not work for short genomes. It is only working for longer genome. And this hyphen ADIV, it does not work for long genomes. It is only working for a shorter genomes. So as of now, even if you are just using a one chromosome, but it's a human data, and human data will never, like, it will not a small data. It is like a longer genome, obviously. It cannot be a small data. So that's why we are using this first option, hyphen A BW TSW. And then you have to provide your input FASTA file, which means your reference genome. So how to prepare this command? You just have to type BWA index hyphen A BW TSW, and then you have to provide your reference genome, so which is like chr17.fa. No need to provide the output file because by this same name, the reference genome is going to create. Sorry, the index genome is going to create. Okay. So I'm just going to copy this command. First of all, I will give it into the chat box. Recording meeting access you will get in the LMS. The YouTube link will provide there. So just check it once there. Okay. So now I'm just going to copy this command and I'm running the indexing step. So you can see that here it is performing the indexing. Are you performing this? Yes. If you are performing, then I will wait. Means okay. What about others? Just tell me. Otherwise, like I will wait if you are performing. Taking time. Okay, no problem. Maybe due to the internet, it will take time. No problem. What about others? If you have any query, just tell me. When did we create the adapter file? In a fastp command, we created the adapter file. I have shown you like we need to use a touch command for it. It will create one empty adapter file and then we have to paste the sequence which we want to trim there. Okay, Priyama? Okay. Janvi ma'am, what you have to do is first just run this command sudo apt-get update. So can you just run this command, Janvi ma'am, first? And once you run this command, after that, just try to install any one of the tool, either the FastQC, Katada, sorry, FastP, whatever we have given. Uh, Priya ma'am, I have already shared the command in a chat box. Please go through it once. Tell me, Janvi ma'am, if it's working or not. Okay. First, run this update command and then install the fast QC. See whether it is installing or not. Okay. So here you can see the indexing is done, and once the indexing is done, by the name of CHR17 here, some files are created. Actually, these are the binary files you don't need to open, and you don't need to see that what information is present inside this file, because these are the binary files. Okay, so once this binary files are there, once the indexing is done, then the next step, we are going to be align the read to the indexed genome. And for that, we are going to be use the BWA MEM algorithm. BWA MEM. MEM, that stands for maximal exact matches. This is an algorithm of alignment in the BWA. So how to prepare this command? BWA, then MEM, which we are using the algorithm. Then if you want to use any option, you can just go through the manual here and just check out the options and you can use. Then you have to provide your indexed genome, and then you have to provide your input files. So we are having the two input files right now. So let's prepare the command BWA MEM algorithm we are providing. Then we have to provide our index genome. So this is the same name, whatever the name we are having for the reference genome, because tool can by default understand the difference between the reference genome and the index genome. Then we have to provide our input files. What are our input file? It's a trim underscore this trim SRR fastq file. This is our first input file, and then this is the second input file. Trim SRR underscore 2, that is a forward and reverse. And then it is going to give you the output file. So I'm giving a prefix BWA because as we use the BWA just to understand it, then I'm giving the ID, and the extension I'm giving here is a BAM. So it is going to generate the BAM file. You remember what is a BAM file? It's a binary alignment map format. In a very first session, I have told you that this BAM file or a SAM file, it will give you the complete alignment information whenever we are aligning the read to the reference genome. So this is how we are getting the BAM format. I'm just going to share this with you. So I'm sharing this alignment command. And today what I will do at the end of the session or after the session, I will share this script in the WhatsApp group. So maybe whoever is not able to copy paste the command, they can just go through the script and run it. So here I'm running the mapping command. Yeah. Any doubt? Uh, Recording you are getting on LMS, Chetna ma'am. The video link is provided. In the WhatsApp group also they have provided you the video guidance like how to go with how to check the recorded videos, how to enroll the LMS. So just go through it once and you will get the recording there. They are providing you the YouTube link. We are providing you the YouTube link. Okay? Yeah. It's not loading. Uh, okay, just ping in the WhatsApp group, ma'am. Uh, what is the problem? Why you are not able to install the Ubuntu 20, sir? Yeah, for now, if you have Ubuntu 24, that's fine. You can use that. Nikita ma'am, what is the thing that? Okay, that's a very good question and good observation, Nikita ma'am. Like in a fast QC, we are giving a two input files and we are getting two output files. Like for a forward separately and for the reverse separately. But for a mapping, even if you are getting a two input files, forward and reverse, we are getting only a one output file. Why it is like that? Because what we are doing now, we are aligning the read from forward side as well as from the reverse side to the reference genome. But even if we are aligning from forward and reverse, it is going to be aligned at the same position, right? So that's why whenever we are performing the alignment, even if it is a single end data or a paired end data, we are getting only a one alignment file. Okay? Hmm. Save the project. Which project, Noor ma'am? At a time, we can run only one command, Nikita ma'am. Yeah, like you can save the script, ma'am, or the command, ma'am. I'm sharing the command anyhow in the chat box, or like after the end of the session. After the session, I will upload the script in the WhatsApp group as well, so you can save that. Okay, for BWA to BAM. See, like whenever we are running about the command, Nikita ma'am, like we need to run all the commands one by one because sometimes, like if the data is huge, so it is not possible in our normal system to run a multiple commands at a time. Okay, so it's better to run the command one by one there. Okay, all right. So now here you can see that the BAM file is generated. You can see here the BWA BAM file is generated, which is a binary alignment map format. But once the alignment is done, what can be our next step that we need to call the variation? We need to find out like how the bases from the read, they are different from the reference genome at a specific position. But sometimes what happens, like in the BAM file, there are some duplicated reads we are having, and we need to remove the duplicated reads. Okay, because if we are having a duplicated reads, so one read can align at a multiple position, like a same read is going to be aligned. So at that time, we can misinterpret the result. So that if there are duplicates, we need to remove that. And to remove the duplicate, we need to arrange this BAM file into sorted BAM format. Because sorted BAM format means what? Based on the position of the read where it is aligned. Why? Because if the data is in a sorted format based on the position, so like it will give you the BAM file, it will have some positions like the read is aligned at the first base, second base, or a third base. So if we sort this file based on the position, so at that time the tool will able to understand if it is having a duplicated reads or not, and if it is present, we can remove it. So what will be our next step now? We need to do the conversion of BAM into sorted BAM. Okay? Conversion of BAM into sorted BAM. And for that, we are using the SAM tools. Okay? We are using the SAM tools. So I have told you to install the SAM tools yesterday. So you have to install by sudo apt-get. And in the SAM tools, if I will just open the manual, you just have to type the SAM tools, enter, it is going to be open the manual. And here you are having the sort option for it. Sort. So it is going to sort your alignment file. And that's very simple. You just have to give SAM tools, sort, then you have to give your input file, which is your BWA BAM file, and then you are giving your output file sorted. Then here we are giving the option BWA BAM. Okay. So what it is going to do, it is going to sort the BAM file based on the location. Okay, one more important thing. Tomorrow, basically, we required one tool called GATK, and that tool we need to download it by today. You have to go with the GATK download. Okay, GATK, which is we are using for variation calling analysis. Go with the download latest version of GATK, but we are not downloading the latest version. We are downloading the 4.3.0. Yeah, this. So I will share this copy link like I will share this link address, and please download it in your system today only. It's a zip file. So what you can do, whenever the zip file is downloaded, just keep it into the CRISPR folder and then extract it. If you want, then extract it. So it is going to extract one GATK folder. Is it clear to everyone? Yeah, I'm giving the sorting command. Yes. Actually, the latest version, I when I was checking, I I found some problem there. Means like I think they are unstable version for now. So I'm just going with the known version, which is like I'm just going with the stable version, which is known. It's not 4.0, it's 4.3.0, sir. Yeah, please download that GATK by today only. Just kept it for the download, and you can proceed with your commands for now. Okay. Now, I just want to tell you one more thing, one another command, which is completely a optional command, because this is a BAM file we are generating right now, which is a binary format, a computer readable. But if you want to see how each and every read is aligned to the reference genome, so you have to see this result in a SAM format, which is sequence alignment map format, right? Human readable. So just to understand or just to show you, I'm converting this BAM file into a SAM format. This step is completely optional step. Do not generate a SAM file normally for real-time data because the SAM file is a huge in size. It will occupy a lot of space in your system. Okay, so I just want to show you that what columns and all it is having. So let's just go with. So for that, we are again using the SAM tool. So conversion of BAM into SAM using the SAM tool, and I'm just writing here this step is optional. SAM tools, and we are using here the view option because we just want to visualize the BAM into SAM view. So we are providing the input file, which is our sorted BAM file, and we are providing the output file, which is sorted SAM file. So SAM file, it will give you the information in 11 mandatory columns. I will show you. I'm running this command. I will paste this in the chat box as well so that, yeah. And here, if you will do the ls, like you can see this SAM file is generated. So to display this SAM file, I'm using the head command. Head command is used to display the first few lines in the file. I want to display the first 50 lines in the file, and then I'm giving the name sorted, then SAM. And you can see here it is displaying the first 50 lines in the SAM file. And the SAM file is giving you the information in 11 mandatory columns. So what columns it is giving? So the first column. First column, it will show you the read ID of your read, which is aligned. Second column, it is showing you the flag score. These are some statistical score which you don't need to be worried about. Then the third column, it will show you at which chromosome your read is aligned. So obviously, as we have used only a one chromosome, so it's just showing you the 17th chromosome. Then fourth column, it will show you at which base your read is aligned. This is the base position on the chromosome 17 where your read is aligned. This is the first base position. Then here you are having the mapping quality. Then here you are having the cigar value. You can see cigar value is there. Then here the next three columns, it will show you the information about the next read, like to which chromosome the next read is aligned. And if the information is not available, it is giving like a dash star like this. Then at which base position your read is aligned. Then here it is showing you the template length. Then the 10th column, which will show you the actual read sequence. And the last column, it will show you the ASCII characters, which are representing the Fred quality score. So whatever the column I have shared it with you, obviously it's not easy for us to remember. So I'm sharing one PDF with you, okay? So you can just go through that PDF. Um, just a second. Yeah, so I will share this link and you can see here, if you just go down, they have provided all the 11 columns information, like what information we are having in each column. So here, from here, you will get it. I will share this PDF link. Just save it somewhere so that in future, if you need, you will get it. Okay. So now you can see this SAM file is sorted based on the position. These positions of the base, they are in a ascending order only. And you can see there are some duplicated reads we are having, some positions, they are same. This ID is same. See? So in this case, what we have to do, this is just only a first position we are getting, second and third. But there are so many duplicated reads we may have, and that's what we need to just remove the duplicated reads. And for that, we are using the SAM tools again. So what is our next step before the variation calling? That is to remove the duplicates using SAM tools RMDUP algorithm. Okay. If I will just clear this and just type SAM tools space RMDUP on the terminal and enter, it is going to be open the manual. So you can see this, like we need to use these two different commands. SAM tools RMDUP, that is to remove the duplicates. And there are two options they are having. One is a hyphen small s and the capital S. So what is the meaning of small s and capital S? So basically, this option it tells to the SAM tools RMDUP to consider only the sequence of reads when identifying the duplicates. So whenever they are identifying the duplicates, at that time only it should like it is telling to this SAM tools RMDUP to consider the sequence. Means in other words, if I will tell you that it ignores the other characteristics of the reads and only look at the actual sequence of the DNA or RNA that reads represent. Means think like it is only looking at the words in the book without caring about anything else like the page number or author's name, or it is just interested that what words are saying. This is just what example I'm giving. But the capital S, actually, this option it is telling to the SAM tools RMDUP to perform the stricter duplicate removal. Means like it is going to consider both the alignment, start and end position. Then it can be useful in the situation where sequencing errors or variations might be causes slight differences. So at that time, you can go with the capital S. So small s is focusing only on the sequence and capital S is a stricter option. So in our case, we want to focus on sequence and we want it should be a strict also. So that's why we are using both the option, small s, capital S like this. Then input sorted BAM file it needs and then the output file. So we have to prepare the command SAM tools RMDUP small s, capital S. Then we are providing our input file, which is your sorted BAM file. And then you have to provide the output file. So I'm giving the prefix as a RMDUP just to understand. Then this SRRID dot BAM. So this is our command for the removing duplicates. I'm just going to copy this command and run it. Yeah. Now how to see whether the duplicates are removed or not. So if you will see the file size of a sorted BAM file, it's a 28 MB. And for the remove duplicates BAM file, if you will see, it's just 7 MB. So you can clearly see some reads are removed. That's what our duplicated reads. So this is about the mapping and removing the duplicates. And after that, we are having the next step, which is a variation calling. But that we are going to see by tomorrow because mapping is done. And tomorrow we are going to see about the variation calling. So now tell me if you have any questions, any doubt. You can unmute yourself. Tell me, you can share your screen as well if you want. Yeah? Anyone? I'm sharing the attendance link as well. First 15 minutes means like why it was not clear, sir? Yeah, kindly mark your attendance. I've provided the link. Not an issue, sir. Just go with the recording once because actually I have explained the whole step. So it's not possible to repeat all the step again. So if just go through the recording and if you have any question about it, just tell me tomorrow. Okay? So
Ma'am,
basically, now, yes,
okay, sir.
yes,
Can
yes.
you please tell them to update the LMS for recording because we are unable to actually visualize.
Actually, sorry, I'm also not aware about it. That like they are, it is just seen by the management team actually. Uh,
I have informed them, but they haven't any answer.
Actually, they are working from 10 to 7 only, so it's okay. Like I will just inform her tomorrow morning. So maybe if it's not like you're not able to access it from the LMS, she will share the recording like how to access. Otherwise, we will directly provide you the YouTube link. Okay.
Yes, ma'am. Thank you.
Okay. Yeah. Okay. Okay. I will just convey your message so that maybe you will get the links. Uh, this message is already shared in the WhatsApp group by Sona Patil. If you will see that it is showing you introductory video recording is also provided and LMS login link is also provided there. So just go through it once. Okay, Rutuja ma'am. They have provided the password and all as well.
Yeah, but it's showing loading. It haven't been shown like a recording section.
Can you just try it once? Because what happens like sometimes even if we upload on the YouTube, it take more than 1 hour to upload. So that's why maybe when you are checking, it was not uploaded completely. But yeah, because I have uploaded it only on YouTube, and I'm just reaching. I can show you in my system. It is clearly visible.
The YouTube link for which you have provided in WhatsApp, first day recording has been available, but second day is not available.
Yeah, that's what. Like I have uploaded it properly tomorrow. Sorry, today. And like today's session, I will upload it tomorrow. But for the yesterday's session, I've uploaded today only. Uh, yeah, maybe at that time where you are checking, it was not completely uploaded, but now it is uploaded completely. Because I just checked at 4 p.m. only.
Okay, I will just. Thank
Yeah,
you.
yeah, yeah. Just go through it once. It is available now. To run the Ubuntu and Linux, is like if you are asking about the number of processors, at least 16, if you want to deal with the human data, so 16 GB processor you required, ma'am. More than that, if you are getting, that is better, but at least 16. Yeah, we can use any another chromosome, but whatever the data we have selected, ma'am, it is mentioning specifically that we are checking it on the BRCA1 gene. And BRCA1 gene is located on 17th chromosome. That's why I have just taken the 17th chromosome. Okay? Yeah. So please download that GATK tool, paste it into the CRISPR folder, and extract it. So it will extract one folder by the name of GATK. We require that folder because this tool we are going to use by tomorrow. Okay? And I will share the script uh, now only in the WhatsApp group. So just go through the script. So whatever the commands are left, just run till whatever we discussed today. Okay? Uh, Actually, for others, it is visible, sir. So just check it once now because all the recordings are uploaded on the YouTube. And still, if you are facing the issue, just put the message in the WhatsApp group. Okay? Yeah. Okay, so that's it for today. Rest of the things we'll discuss by tomorrow, same timing, 8 p.m. Okay? Till then, bye. Have a great day. Good night.
Thank you, ma'am.
Yeah, you're welcome.